it's not my favorite brewery.
Okay.
I'm not gonna say I hate it. That's
We're just gonna do,
really.
Yep, that's the way to do it with the PowerPoint directly.
Oh,
Yeah,
it's
because if you do your desktop, you'll get all sorts of extra crap.
okay,
I've never
does
been
that look
there.
okay?
I feel like this is the third time we've seen it.
Yes.
They're very good.
I would like to know what the bug on the next screen, second from the left on the bottom is.
Yeah.
Okay, is it all, does it look correct? You're seeing the slideshow now?
That's online. Yeah, we're seeing the slideshow.
I like, they have the watermelon one.
I went to the wrong
It's
classroom.
they fly
You have water?
pictures there's the watermelon.
Sit over here.
Oh. Did this take the wrong friends?
You went to the wrong classroom.
Conehead, Green State Lager.
I don't want to intrude.
That one's over there. Let's go.
Pretty good. You see everybody clusters in the back. So there are more people that are joining on, and I think, you know, because of the weather, there's a fair number of people that will,
Religiously at 4 o'clock. every day.
Okay, Lori's here to get started. Okay, we're gonna get started. It's my pleasure to call Ginsberg, who is just months away from completing his Ph.D. At University of Georgia in the Department of Genetics. He will talk to you more about his current Ph.D. thesis, but he's really interested in looking at things like evolutionary processes and speciation, and especially looking at kind of endosymbiote mediated processes that contribute to speciation. So Paul has been active in working in Drosophila for a little while now. Prior to his PhD program, he was a student at UC Davis, where he worked with Michael Torelli, working on evolution and ecology, and looking at kind of speciation processes there as well. Looking at things like cytoplasmic ability. He's published now in Molecular Phylogenetics and Evolution, Journal of Evolutionary Biology, Current Biology, and he's been active in winning a number of research boards and fellowships. He has a strong track record of undergraduate mentoring at both these institutions. So he's currently also an applicant for a PhD, postdoc position in my lab, looking at epigenetics and the role of epigenetics in rapid evolution to insecticide resistance. So thank you for coming here, especially given kind of COVID, weather, and all the other constraints. We like to kind of put every candidate through the ringer here, especially with the weather. But okay, hope everything's okay, and take it away.
All right. Thank you for the introduction. Thanks for having me. I've had a great time walking through the snow a little bit today, which I don't get a lot of in Georgia. I'm just going to get started. So, as you already heard, my name is Paul. I'm in Kelly Dyer's lab at the Department of Genetics at University of Georgia. Today, I'm going to talk to you about some of my dissertation work, looking at the microbe interaction and maintenance of species boundaries. And so, I'm just going to start with some of my general interests. I think as someone who considers myself an evolutionary geneticist, interested in the fact that organisms are so incredibly diverse, kind of a lot of different ways here. And a driving question is what generates all of this diversity. And so I kind of like to think about it through, so another way to phrase this is what drives and maintains the evolution of independent lineages. I like to think about this as kind of a combination of, relatively speaking, short-term processes, like the development of population structure, local adaptation, shifting migration patterns. Obviously, this is not an exhaustive list, but generating kind of these long-term consequences. Again, not exhaustive, but some things like speciation, extinction, and ultimately study diversification to generate all the diversity that we observe. And when I'm talking about, so let me stop right here. my dissertation research primarily focuses on this process right here, speciation and the completion of that process. And when I'm talking about speciation, I am talking about the evolution of reproductive barriers. I generally think about it in this kind of classic Jeffords-Molar incompatibility context, where you have an ancestral population, a gruesome event becomes isolated. When those populations are isolated, you get fixation of different alleles, especially when they come back into secondary contact. There's some form of reduction of fitness of those hybrids. And so a driving question is what produces these incompatibilities that can evolve in these different populations, and encapsulates all of the major evolutionary forces that I like to think about. Obviously, on a fundamental sense, they all apply to input of mutation, genetic drift, so the stochastic fixation of alleles, which can happen especially when you have really big population contractions during isolation events. Natural selection, which in this case, I'm really talking about local adaptation of these different populations that could experience different environments in isolation. And then lastly, migration, which can, if there is migration between these populations, can impede the process of speciation, or the lack of it, which could enhance and increase the rate of that process. And an important aspect of this reduction in fitness is that it can work on different stages of the production of hybrids. And so we like to break it down into these pretty big distinctions. The first being pre-mating isolation, where individuals from different populations or different species would be mate with one another. And then post-mating isolation, which takes several forms. So both of them are after copulation. You can have either pre-zygotic isolated barriers where the cellular fish from the fertilized eggs. And get a little block out here, where you've got post-zygotic isolation, where hybrids form, but they're either viable or sterile, which we consider intrinsic, or extrinsic, where they are where hybrids are produced, but they're somehow poorly adapted for their environment. And so this can take, take a thing, a myriad of different forms. Somehow they're not well suited and don't fit the fitness optima of the two parents. And you can imagine that any of these processes occur, this becomes a very costly behavior to engage in. And so you can think of, could conceive that the cost of this could indirectly select for pre-mating isolation between populations. This is something that we call reinforcement, which just as a definition is basically the selection against the formation of hybrids in areas of secondary contact. And here I'm showing you something that's very similar to that first model I showed you. We have the barrier form populations in isolation. If there is some gene flow between these populations that can produce low fitness hybrids, that would presumably select for higher mate discrimination, such that you reach a certain point where you don't have hybrids forming anymore, and then there's pre-mating isolation between those two populations. And so, again, the driving question is, what is generating these incompatibilities that can drive this process? And I focus on one in particular that I think has been appreciated more in the last few decades, which is host-microbe interactions. And for the context of this talk, I'm talking about host-microbe interactions, talking about interactions with Wolbachia. Wolbachia is a particularly interesting one because it is pretty strictly maternally inherited. It's cytoplasmic, so you can think of it as being inherited basically like mitochondria. And it's also everywhere. It's super common, and the estimates range a lot, but something on the order of 40 to 60 insect species are harbor Wolbachia infection. And they're particularly well known for manipulating the reproduction of the hosts. And they can do this in a number of different ways, a couple of which I'll describe in detail. But all of them are attributed to the fact that Wolbachia can kind of leverage these manipulations to affect the frequency in their host populations. And it does a lot of other things too. So this is kind of a thorough all sentence. There is a number of different Wolbachia-induced phenotypes. Some of the most common ones that were reported are things like protection against other pathogens, and things like nutrient provisioning, which in some cases has gotten to the point where they've essentially become obligate, so they can provide essential nutrients that the hosts have lost the capacity to produce. A good example of that is the Wolbachia infection of bed bugs, or an obligate Wolbachia infection. This is just a fun picture of Wolbachia. So this is a cell of Drosophila lugula, a different pleasure-feeding Drosophila species, and all of these green dots are Wolbachia inside the cell. This is not a particularly important point, but you can tell that this Wolbachia infection is really concentrated in the cell. That's another thing that can vary a lot too. Often has implications for how Wolbachia affects us. Sorry, I have to jump forward in slides because I don't have a way to see what slides come next. So apologize. I'll try to limit that as much as possible. An interesting aspect about Wolbachia infections, likely in some part due to these reproductive manipulation, is that they can fix really rapidly in populations. And so this is just an example of this, where I'm showing populations of Drosophila simulant across the eastern coast of Australia. There was a resident moderate frequency Wolbachia infection here, and over the course of only 18 or so years, a novel Wolbachia infection started to show up and then completely displace the other Wolbachia infection and spread the high frequency. And so this is one example of this, but we have a number of examples of this across many different insect species and insect orders. And as I said, largely or in part, this is explained by the fact that they manipulate reproduction of the host. Oh, geez, I'm sorry. Okay, I don't see my slide. So what I wanted to say next here is that this is a very rapid process, and we can see the consequences of these sorts of processes happening in our lives. And so this is part of work I was involved in these labs. We're essentially trying to look for patterns of evolution, coevolution between hosts and their Wolbachia infection. So here on the left, I'm showing you a phylogeny of several different Drosophila species. And then on the right, I'm showing you a phylogeny of their Wolbachia infection. And there's really two things that take away from this figure. The first, a high degree of topological discordance between the host and their Wolbachia infections. And secondly, if you look at this root, this node here on the host phylogeny is estimated to be between 10 to 50 million years old. Whereas you look at the analogous root on this Wolbachia tree, we're looking at about on the order of 5 to 30,000 years old. And so this tree is much, much shallower than the host tree. Both of these things suggest that Wolbachia moves between hosts much faster than host species. Okay. And the question is, why does this happen? And it's at least in part explained by some of these reproductive manipulations. The most common of which we call cytoplasmic ability. And something happened with my formatting here, but basically, if you think of all possible crosses with infected females produce infected offspring. Two uninfected individuals, which again happened with the formatting here, produce offspring. But this last cross out here, which is supposed to be an infected or an uninfected female with an infected male, results in some level of embryonic mortality. Okay, obviously hard to see here because it's not showing what's supposed to be, but just remember that one cross is really easy to know. Uninfected female, maybe to an infected male, results in some level of offspring mortality. We have a pretty good theoretical framework that can explain how this can affect its frequencies and population. So essentially described by this equation right here, where P hat is equilibrium frequency in adults. And then these three parameters, the relative fecundity of infected female, mu, the frequency of unaffected ova produced by infected females. And because this is basically a measure of the maternal transmission rate. And lastly, S subscript H, which is a measure of that side effect. So the relative reduction in hatch rate of the incompatible cross compared to the compatible cross. And if you squint at this just in the right way, you can see that this is essentially the quadratic formula. And so there's two solutions to this equation, depending on the sign of the root that you take. If we take the positive root, we get a stable equilibrium frequency that tends to be quite high in 1. And if you take the negative root, we get this unstable equilibrium frequency. What this model tells us is that if we have an initial infection frequency below this unstable equilibrium frequency, we'd expect that Wolbachia infection to be selected out of the population. If we have an initial infection frequency above that unstable equilibrium, we expect CI to take over and to push that block infection frequency up to a stable equilibrium. A kind of simple way to think about this is that this frequency right here is the frequency at which is enough infected males in the population that it becomes disadvantageous to be an uninfected female. Once that point is reached, CI pushes that infection frequency up to the stable equilibrium. Okay, and so, sorry, I keep jumping forward. I'm going to shift gears a little bit here. I told you about speciation and Wallachia, and now I'm going to go back and tell you a little bit about the system that I studied and incorporate these two aspects. And so my system, I study the Drosophila Sublimaria species complex, which is comprised of 3 species. These are all mushroom-feeding Drosophila species. In teal, I have Drosophila transversa, which as far as we know is an old world species. But I have question marks right here because we don't know if they both occur in these areas. As far as I know, there is one record from 2009 of Drosophila transversa being collected in the Kamchatka Peninsula, which is eastern Siberia. That's all I know. But I'm primarily going to focus on these 2 new world species, Drosophila recens, with this range highlighted in orange, and Drosophila sublimaria in blue. And you can see they share a range of sympatries, right, which is basically the Canadian Rockies and about 1,500 kilometers. And so when you have a pretty good just characterization of how these 3 species are isolated from each other in the lab. If you cross either sublimaria or transversa recens, you get a pretty kind of classic Haldane rule pattern. Males are all sterile, females are fertile. When you cross sublimaria and transversa, that's not true. They produce perfectly fertile males and females. When so my formatting here got messed up again, but this is supposed to be the female sublimaria across the male recens. You have very, very strong mate discrimination. So, and this is specific to sublimaria from this part of the range where they overlap or they're in symmetry. Whereas with all the other crosses, we have kind of moderate mate discrimination. So this is kind of indicating that reinforcement is likely playing a role selecting for, in this case, just sublimaria females to discriminate really strongly against these males. The reason we think that's the case and that this isolation is asymmetric, or the level of it is asymmetric, is because we have a Wallachia infection that is essentially fixed in Drosophila. And so, again, I'm just zooming in on this part of the world here. These 2 species, recens in orange, fixed for a strong CI causing Wallachia infection. And sublimaria in blue, which doesn't harbor any Wallachia. And so, just to reiterate what I said before, only this direction of cross with an uninfected sublimaria female to an infected recens male, you get essentially complete embryonic mortality because of that CI effect. The reciprocal cross, we don't. So they produce hybrids. Males are sterile, and they're probably a little sad, but otherwise fine. And so our first question when we look at this system is, how are these species related to each other? We have pretty good predictions on what we expect to see based on the recent post-zygotic and pre-mating isolation. And what the sort of genetic pattern that we can detect that explains that can explain some of these phenomena. And so I'm going to walk you through a few phylogenies now of different regions of the genome between species. And so first I'm just showing you phylogeny generated with 2 Y nucleotides, the 2 fertility factors, about 2,500 base pairs. And then I'm showing you all of the same species here. I've just broken up recently into the region that's sympatric with sublimaria versus allopatric. And then similarly, I've broken up sublimaria into sympatric and allopatric population. What we see is pretty much what we expect here. Right? So we have recens grouping as an outgroup to sublimaria and transversa, which is consistent with what we see because recens cross with either of these 2 species generates hybrid males, however these 2 don't. So this is pretty consistent, and we wouldn't expect there to be any Y chromosome regression across the boundaries or anything like that, because now they're sterile. When we look at the mitochondria, it's similar, but slightly just slightly different. So this is just a phylogeny of a single mitochondrial locus to a one, showing you all the same species and populations. Again, we see regions as an outgroup here in red and orange. And then we have my two primarily sympatric Sublimaria and allopatric Sublimaria populations, although there is an intermixing between these two. And then transversa down here. And this effectively forms a polyphony. There's pretty low support for either of these branching as more closely related to one another than the other one. What particularly stands out here is you can see there's one sample right here. This is a Sublimaria sample, surprisingly from an allopatric part of the range. That's either from Seattle, who was collected it. It clearly has a recent mitochondrial haplotype. This is we made a single observation with this data set. This is something that we observed pretty much every single time we sample Sublimaria, on the order of about 4% of Sublimaria individuals have a mitochondrial haplotype that looks like it definitely came from Drosophila. And so now I'm just going to showing you a nuclear phylogeny, where you see there's a couple differences between how these species are related to each other. This is comprised of 27 nuclear loci, 18 of which are autosomal, 7 of which are X-linked. This actually also includes the 2 Y-linked loci. As you can tell, or as you might expect, if I remove these two, it looks pretty much the same. The Y-linked loci would give a very different signal, so this is despite the fact that these two are included. And I have the same groupings of my different species. So here, looking at recents, which is essentially intermixed relative to the two populations. Then I have my Sublimaria populations, sympatric and allopatric, and Drosophila transversa. So absolutely, there's a shift in how these species are related. Now, transversa looks like it's now for species. Allopatric and sympatric Sublimaria are split. And I think most interestingly is that Sublimaria, that is from the sympatric part of the range, now looks like it is paraphyletic and sister to Drosophila. A pretty major, seemingly non-random shift in how these species appear related to each other. And so the next question, what question this brings up is what produces this change in the phylogenetic patterns that I see, depending on which part of the genome I'm sampling. And really, when you're trying to look for phylogenetic causes of phylogenetic discordance, really comes down to these two phenomena: incomplete lineage sharing, which important distinction is a stochastic process, or gene flow, which is not a stochastic process. Just as an example of that, I'm just showing you a species tree outlined in black, and then segregation of a locus in blue here. You might expect that the majority of the genome would be comprised of loci that reflect the species relationships. However, if you have ancestral variation and rapid splitting of lineages, showing you just a couple of examples up here, you can have alleles randomly segregating in ways that don't reflect the species phylogeny. Okay? And so here's just two different examples, but importantly, because this is a stochastic process, you don't expect any to show up more frequently than the other one. Kind of expect all of discordant pathologies to show up in similar frequencies. Gene flow, that's not the case. The gene flow is not stochastic, where the specific lineages that are exchanging genetic information and the rate and magnitude at which they do that will give you an enrichment for specific discordant topology. And just an example of that here. Here's gene flow between these lineages. We expect an enrichment specifically for this discordant topology. And so I can do this with my multi-locus data set. And here I'm just I'm using a test, a pretty old conservative common test that people use to distinguish between these two possibilities, called Abba test. And so I'm restricting my data set down to sites that are biallelic, where 2 of my taxa have one allele and 2 of my taxa have another allele. You can imagine that if there was a mutation somewhere along this trait, you would see the distribution of these 2 different alleles in a way that's according with your species phylogeny. However, if you have mutation on an internal branch, you will get one of 2 possible sites. You have gene flow between these two lineages, you get an abocyte. And if you get these gene flow between these two lineages, you get a babocyte. Again, if this is an effect that is the result of include lineage sorting, we expect these two disparate topologies to show roughly equal in roughly equal. So I can apply this to my data here where I have my allopatric subplanaria, sympatric subplanaria, regions, and I'm using Drosophila corneria as an outgroup here. And when I count the sites in my data set, it's kind of a limitation here. I get 28 abocytes and 10 babocytes. So the limitation is from the data that I have, which is this multi-locus sequence data set. And the fact that I'm using basically a consensus sequence from all of my axes here. So I'm not able to get data from the polymorphism when I use this method. Nevertheless, this shows an excess of abocytes relative to babocytes. This is just the test statistic that uses testes, and it's a significant result. And so the excess of abocytes suggests that there is, in fact, gene flow going on between sympatric subplanaria and Drosophila. Another way I can get a handle on how gene flow is occurring between these species is modeling it in a coalescence framework. So the one thing that ABAP test doesn't do is it doesn't give you any idea of the direction of gene flow. It just tells you whether or not there's evidence for it or not. So to get an idea of what direction this gene flow might be occurring, I took my autosomal data and ran coalescence simulations that give me estimates for migration rates from regions into subplanaria and subplanaria into regions, and estimates of which is essentially my effective population size. An idea of the population size of the organisms. And I ran 4 models, one that allows gene flow in both directions, one only regions into subplanaria, one only subplanaria into regions, and the one that assumes that gene flow in these directions is symmetric. And when I do that, the most supported model using log base factors is a model that allows for gene flow in both directions, but primarily in the region going from regions into subplanaria, roughly 3 times the rate. So I should mention that these estimates here are mutation scale. So you can get a kind of an approximate estimate of how many migrants per generation by basically just multiplying these 2 numbers together. When I do that, the estimate for regions going to subplanaria comes out to about probably a few migrants a year. Okay. And so I started this section describing how Wolbachia in Drosophila recens, which is essentially tick, causes strong CI and can help contribute to the maintenance of the species boundary. And now, what I just showed you indicates that, or suggests that we have gene flow going primarily in the direction of recent by infected species into subplanaria by uninfected species. And interestingly, this includes clear signs of mitochondrial progression, which is interesting because, like I said before, Wolbachia is inherited just like mitochondria through the maternal grandma. And so the question is, we know that CI causing Wolbachia infection have this pattern of spreading quickly through populations of different species, and this seems like a system that is really ripe for that to occur. The question is, why don't we see that? So to answer this question, I generated Wolbachia infected Drosophila subplanaria in the lab, which is a pretty simple process that takes a little bit of time. Cross-infected female regions to uninfected male subplanaria, fertile infected F1 hybrids, and then back cross into subplanaria for 10 generations. So effectively replacing the genetic background of regions with subplanaria, but carry along Wolbachia and mitochondria. And then I do this annoying but simple process of creating an uninfected copy by treating with tetracycline. So I take tetracycline, soaked mushrooms, let flies ovipositor for 5 days. Yeah, I say annoying because flies apparently are not very happy when they only have tetracycline soaked mushrooms to oviposit on. And then roughly 14 to 20 days later, which is approximate generation time of these species, I can collect newly emerged flies, reconstitute their microbiota as best as I can. So basically what that process entails is just taking dirty food that I let virgin flies kind of feed on and stuff, and then take them out and let these flies feed on there for a little bit. So we can pick up any kind of opportunistic infections that are just common in the system of the species. And then I can test for Wolbachia infection, which is just a simple PCR assay. And so now I have my infected and unaffected copies, and I can use that theoretical framework I talked about before to estimate my stable equilibrium and my expected unstable equilibrium frequencies, which is just estimating these three parameters: the strength of CI, relative fecundity, unaffected versus unaffected females, and then maternal transmission. And so now I'm just going to walk you through the estimates of these three parameters. And so the first thing I see is that, like we already knew, strong CI and Drosophila recents. So what I'm showing you here on the y-axis is the hatch rate. I'm going to show you my two cross types: uninfected female by infected male, an incompatible cross, and uninfected by uninfected. What you can see in both cases, recents in red, subcanary blue, is that my incompatible cross is a much lower hatch rate than my compatible cross, and that appears to be a stronger Drosophila recent. Also probably notice that there's a bit more variation in this Ginsberg line. So what I can say about this, I don't have a slide or anything, but so I did this backcrossing experimentation with Sublimaria from throughout the range. So I wanted to sample from Sympatric and Allopatric population, and this effect is not this effect is consistent across this genotype. So I think it's likely more a case of either some mitonuclear interactions or something like that. But nevertheless, for the most part, I get pretty high hatch rates in these crosses. When I look at fecundity, I really don't see any difference at all, which is kind of surprising. So kind of a priori assumption is that being infected with something at a high level costs something. But as far as I can tell, it doesn't appear to be the case. So again, I'm showing you recents in red, subcanary in blue. First column is uninfected females, second is infected females, and I'm just showing total offspring production. You can see in both cases, there's no there's no difference between the two. And in fact, so these are showing medians, but I'm only really concerned with the means. In fact, in both of these cases, infected females tend to have slightly higher fecundity than uninfected females. It's something that has been reported before, although it's relatively pretty rare. I don't have a figure for maternal transmission just because it's a number, but basically I take infected females, I make them to uninfected males, and then I collect all of those offspring, screen them for Wolbachia, see how many uninfected offspring are produced from infected females. And I can put all these numbers together and estimate my unstable equilibrium frequency. So here I'm just showing my estimates for CI, for fecundity, U, and my 95% confidence intervals, and three frequencies there. And then I have my unstable and stable equilibrium frequencies with what are essentially upper and lower bounds for that estimate using combination of parameters in that 95% confidence interval. And something kind of interesting that pops out is that in both cases, excuse me, the unstable equilibrium frequency is 0. And so why is that the case? Well, if you imagine at very low infection frequencies, there's not a lot of infected males in that population, and so CI doesn't really matter that much. At very low infection frequencies, you're really only concerned with the relative fecundity and the production of uninfected females, or uninfected offspring from infected females. If these combined parameters are above 1, with this deterministic model, that Wolbachia infection will spread. And that's true in both of these cases. So really high maternal transmission and a high relative fecundity in both recent and Sublimaria produces an unstable equilibrium frequency of 0. Obviously, the lower bound is 0, and the upper bound in both these species is right around 10%. So conceivably, the actual estimate is at 10%, and that's why we don't see Wolbachia invading. But I expect it more likely to be closer to my point estimate, which would mean that given just this, I expect Sublimaria to be invaded by Wolbachia. But again, we don't see that. So the question is why? And as I mentioned earlier in the talk, Wallachia does more than just this. It can do a lot of different things. As a way to take a kind of exploratory approach to address this, I did a differential expression analysis in some area where I have 3 biological replicates of infected females, 3 biological replicates of uninfected females. Each female, I pulled reproductive tissue and retained the carcass tissue. And just to be clear, I pooled samples of individuals here, it's not just a single individual. Checked RNA, do 150 days for parallel with the sequencing. And I run it through this pretty straightforward pipeline. I did de novo transcriptome assembly in Trinity. You don't actually have reference genomes for any of these species. I'll talk a little bit about second. Transcript quantification with salmon. And I did my differential expression at edge R. And I'm primarily concerned with these 2 comparisons. The differential expression between infected and uninfected reproductive tissue, and same thing in carcass tissue. When I first look at this data, I see that a very small percentage of my variance is attributable to Wallachia infection. And so here I'm just showing a PCA of all of the quantified transcripts. And basically my first principal component here represents tissue, my carcass tissue on the left, ovary tissue on the right. And my second principal component is essentially represents Wallachia. So both of my uninfected tissues up here, both of my infected tissues down here. But again, a very small portion of the variance, only 1.7%, which is surprising. And so when I test for differentially expressed genes between my infected and uninfected tissues, we don't see a whole lot. This is a real bummer. Honestly, if you put all of the genes in this organism on a dartboard and just threw darts, I feel like it would probably give you more signals than this. So I only saw 56 upregulated genes in my carcass tissue, literally 1 downregulated, and then similarly small numbers with my ovary tissue. And they didn't really tell me a whole lot. And I've been trying to figure out what is going on with this analysis, and I think a lot of this is likely due to the fact that I'm using de novo transcriptomes. So kind of as a rule, de novo transcriptomes, you get an unrealistically high number of predicted transcripts. These are really permissive algorithms, and so which could diffuse signal during quantification. It's one potential solution. A completely different part of my dissertation I'm not going to really talk about today is generating these reference transcriptomes or reference genomes using those to map my RNA data. So that's going to be in the future. But overall, this is a real bummer and very surprising. So let's move on. Another possibility is that this Wallachia has not been along for a very long time. So everything I've showed you so far is using a recent sample that was collected in 2015 to generate my infected Sublimaria sample. So through discussions, she suggested I try this again. And now I'm doing the exact same thing, except I use the Drosophila recent collection from 2001, roughly 15 years earlier. So exact same thing, except now I'm using old Drosophila recent. Why would I do this? Kind of what I described in the background. We know Wallachia infections can rapidly replace other Wallachia infections. And so it's kind of maybe a little bit of shot in the dark. But I do that and I get a pretty interesting result. So again, with my 2015 recent collection, which causes CI recent, also causes CI in the area. When I use my old recent collection, it also causes CI in Drosophila recent. It causes male killing in Drosophila area. Male killing is another relatively common reproductive manipulation that will lock in by host. And it's basically exactly what it sounds like. Affected females produce all female brood and roughly half of the offspring. So presumably males are dying either embryonically or larvally. Okay, so the question is, how are these Wallachia strains different from one another? Because, like I said, I'm doing this with all the exact same Drosophila subfamiliary background. So really the only thing different is Wallachia and Cytoplasmic, which I'm going to do more comparative. So we can talk about it more. How do we talk about that? But anyway, to answer this question, I got DNA extractions for both my samples. After nanobore sequencing, I do myself in the lab, and then I send out part of that sample to do the Phylogeny sequencing. With my long reads, I can do a de novo assembly, and then I blast all of my contigs anything that hits Wolbachia, I take out, and I map my long and short reads back to Wolbachia, those Wolbachia configs. And then I use both of those data forms to do kind of a high range of assembly of unicycler. I've done this several different infected samples so far. Pretty reliably generates a whole one config Wolbachia genome, which is really nice. And I can align those genomes in all. So when I do that with my Wolbachia strains that called CI or Melkin and Sublimaria, it looks like this. So on top I'm showing you my Melkin Wolbachia strain. On the bottom I'm showing CI. The different colors represent blocks of homology. And everything above the center line is in the forward direction. Anything below it is in the reverse orientation. And then lastly, any white areas represent sequence that's present in this genome that is absent in the other genome. And the first thing that stands out is that these Wolbachia infections are remarkably similar. So when I look across the genome, I see literally 54 single nucleotide variants. So very, very similar. And I have these two big structural variants. First in yellow, this inverted region, which is about 34.5 kb. And lastly, this region that is present in my male killing strain, absent in my CI causing strain, that is about 25 kb. And interestingly, this is all prophase related. It's a prophase related region that encodes approximately 31 genes. What's interesting about this is that there's in the literature hypotheses about what both are sufficient to cause Melkin in the host. What people have concluded is that it's a locus that falls right about here that is identical between my two Wolbachia strains. And so clearly there's something going on, either a mutation or something in this region here that is either a modifier allele or is itself a sufficient allele that's sufficient to cause Melkin. And I can walk through, I'll briefly walk through some of these. So what do my mutations do? I can break them down into these 4 basic categories. 26 of them, almost half are synonymous, so fall within a coding region, but don't actually change the amino acid sequence. 15 of them are intergenic. 7 of them are non-synonymous hypothetical proteins. Not much I can do with that at this stage. And then 7 of them are non-synonymous changes in proteins that I actually have pretty competent annotations for. And those I'm just listing right here. And as far as I've looked into this so far, I don't see any actual documentation of Wolbachia-induced effects around these genes, although these last 2 are actually pretty good candidates. So in a completely different system, this gene, GID-A, when it is deleted from a salmonella strain, it essentially eliminates pathogenicity in mice that are inoculated with them. So again, completely different system, but an association with pathogenicity, something already relevant. And then second, this oxidoreductase activity in Wolbachia has been hypothesized as a way for Wolbachia to essentially interfere with a host immune response. When I look at this prophage region that's missing, I can do the same thing. I can break it down into these 4 categories. Almost half of them are hypothetical proteins, so it's not going to need RNA data to really get a sense of what's real and what's not. This will just rely on computational prediction. I get 5 anchor and repeat domain proteins, 7 that are just prophage structural genes, and then I get 3 transposases. Some of these, the ones that are actually kind of interesting, are anchor and repeat domain proteins. People have been interested in these for a while because they're relatively rare in Wolbachia, and so are good candidates for protein interactions with other eukaryotic proteins. And transposases, which are just interesting because they lie right in my flanking region. So we're presumably involved in some way with the production of this missing region in one of my Wolbachia strains. Also, I'd say flanking my inverted region too. Okay. So despite those changes, like I said, these Wolbachia strains are incredibly similar. And this the CI causing Wolbachia strain almost certainly arose within Drosophila. And so here I'm looking at a phylogeny I generated using MLST loci, multi-locus sequence typing. That's really common for people to use to look at the relationships between different Wolbachia infections. And this is including a number of different mosquito species, a series of different Drosophila species that I found might be closely related. You see these 2 WREC strains are right here, virtually identical. Okay. a couple of the steps I described earlier fall in one of these, a couple of these ones. It's almost certainly rose within these. However, a similar milk is actually a recent Phylogeny replacement. So there's a potential obviously that I just didn't happen to sample this Phylogeny strain from the ones that I used from my more recent collections. So to answer this question, I've designed primers around the deletions, and what I plan to do after I get back to Athens is to start screening my different geographical collection from different parts of the range and over the last. These are ones I got this in a table collection, but these primers actually work pretty well so far. As far as I can tell. Okay, but either way, this represents, I think, a really great example, really rapid evolution associated with Slovakia and how it can affect its host. I hope everyone's ready for this transition here. Yeah, almost exactly like you. Not exactly, obviously, but humans are also a really powerful agent of rapid evolution. So I'm going to talk a little bit about the work I plan on doing here. Humans are really good at a lot of things, a couple of which being disrupting habitats and fragmenting populations. And on the other hand, we're really good at homogenizing habitats. The best example of that is agricultural fields. And as it turns out, as the name might suggest, Colorado potato beetles really like big potato fields. So they're native to Western and Southwestern America. Mid-1800s, they shifted, some populations of the beetle shifted from their native host plant to domestic potato. And these we've seen since of this kind of outrageously rapid range expansion in the last 150 years. And now they've invaded all of North America to the eastern seaboard, Europe, all the way continuing east into China at this point. So like a pretty amazingly fast range expansion. We haven't only increased the amount of habitat they have at their disposal, we've also exposed them to a lot of different selection pressures. Likely one of the strongest ones being pesticides. They've been exposed to virtually all classes of pesticides. At this point, they've evolved resistance to roughly over 50 different compounds, with a turnover rate of about 10 years since so within about 10 years since introduction of the pesticide, they evolved resistance. You can see that there's just the reported resistance cumulatively over the last 50 or so years. So really rapid evolution of resistance against these pesticides. And the question is, how do they develop resistance so quickly? Again, this is not an exhaustive list, but you can break down two primary ways that this could happen. The first is selection on standing genetic variation, which these two papers, I think, pretty clearly show what's going on at some level. This is actually, from discussions I've had, this is actually surprising to me. You have a really rapid range expansion, you expect a pretty severe reduction in the amount of standard variation, and you don't really see that in the species. If anything, the reduction is modest, which is, like I said, very surprising, but nevertheless the case. And then one that people have thought about more recently is epigenetic modification. And so the Chen lab has looked at this here, so what are they? Where they expose larvae to the pesticide imidacloprid. I hope I'm saying that right. And what they showed is that it was able to induce differential methylation, some of which was in resistance-associated genes, presumably in a way that affects expression of these genes. And I think the most interesting is that this reduction in methylation across the genome was maintained into F2 generation. And that's what I'm just showing you here. The treatment generation, you get high in the treatment, reducing global methylation relative to a water treatment. And then here, the same thing in the F2s. This is interesting because maintenance of methylation profiles doesn't just happen. There has to be a mechanism by which that can occur. Part of the work I'm excited to take part in here is first extending a similar experimental design out to a third generation to see if that methylation profile is maintained transgenerationally, and also associate it with actual phenotypes of with respect to pesticide exposure. But like I said, the maintenance of this epigenetic profile doesn't just happen. There are genes and proteins required for that to be maintained. These are collectively referred to as DNA methyltransferases or DMFTs. They're responsible for making it. I think an interesting observation is that beetles are only one of 4 insect orders that people have actually documented and maintained a complete set of DNA methyltransferases. This is like on the opposite side of the spectrum, Drosophila, the world that I live in. For a long time, people thought Melanogaster didn't even have DNA methylation. Now we know that it happens at kind of important stages early in embryonic development, but they are missing very key DNA MPs. And so an initial question that I have is, what do DNA MPs look like in Colorado potato beetle? And I don't know anything about this yet, but my interest, the reason I think this is interesting is because when we look at different populations of Colorado potato beetle here, you can see that some of them are resistant to imidacloprid, I hope I'm pronouncing that correctly, and some are susceptible. We have a variation in this ability to respond to the introduction of a pesticide, which could presumably be due to variation in these DNFTs. And so I think I'm going to stop there. Thanks everyone for listening. I don't know how I'm doing on time, because that's exactly when I started. But I just want to say thanks to my PI, fellow grad students in the Dyer lab, a particular lab tech who did generate some of the phenotyping data. I've had a bunch of undergraduates help me throughout my years at the Dyer lab, my committee, and my funding.
You can take questions here, and then I'm just going to help you look online.
Sure.
Really, really interesting. So do you think it matters if you are a male killer or you are a cytoplasmic incompatibility version of Wolbachia with regard to reproductive isolation and speciation? Like, is it an aha that, oh my God, this could have been male killing and therefore it should not have crossed like it would for a CI version?
So the dynamics of Wolbachia invasion for male Wolbachia infection are very different, and there's a much tighter range of conditions that you can expect it to spread, and it is self-limited, right? Because it's going to eventually will cause extinction if it gets up to a high enough frequency. So in that sense, I do. As far as overall for the maintenance of the species boundary, my sense is that they are sufficiently diverged, but I don't think invasion of Wolbachia would change anything too drastically. I think things like the production of sterile males is sufficient to maintain, although you can still get a lot of yeah. I don't see, of course, anything's possible, but I don't see that going away, irrespective of the Wolbachia infection. Something I didn't talk about that's actually really interesting is the fact that Wolbachia infection seems to have produced a high rate of mate discrimination has actually cascaded to the point where those sympatric subplanary females don't like to mate with sympatric individuals from the allopatric population. If you had that CI Wolbachia infection spreading through subplanaria, I can see that breaking down. Yeah, but between subplanary reasons, probably.
I think it's always so fascinating to kind of look at kind of these structural variation and kind of how it functions so differently and everything like that. I was just wondering, do you know if there's any way sort of these, do the equivalent of something like RNAi that targets particular things within Wolbachia to kind of see if you can kind of see these other phenotypes and stuff?
Yeah, so there are labs that do that. Address that exact question. Gene editing in Wolbachia has thus far been unsuccessful, or as far as I know, at least in the published literature. But systems that people use are essentially take Wolbachia genes and express them in the host. That one candidate that I talked about that's in the literature for producing male killing, that's essentially what they did. They took that locus, they put it in a and looked for whether it caused no kill. That's also the same way people figured out the genetic control of cytoplasmic incompatibility, which is a pretty well established one. Is you just express it in the host. That's kind of the best we can do at this point. People have tried RNAi and things like that. Kind of unsuccessful. Yeah.
Yeah, no, it'd be just kind of cool, especially with your 7 targets.
Right. Yeah. I'm not a molecular geneticist, so this is the kind of thing for the next graduate student. Yeah.
And then on that note, I
Yeah.
have a question about your initial phylogenies.
Sure.
You made a phylogeny with one loci,
The mitochondria.
right?
Yeah.
I have an issue with that. So
Yeah, I
how
understand.
informative is that? How inbred are the insects that you use, and how
Yeah.
would that influence like
Yeah.
the phylogeny? Like,
So-
so why wouldn't you just sequence the genome's really small? Why wouldn't you just sequence the genome and then build a phylogeny?
Yeah, so that's one of the things I plan on doing. Like I said, I'm generating genomes for these species, and I haven't done in the process of getting those analyses put together. I was working with the data set I was given. It is my short answer, and that I had available to me at the time. So I was dealing with a multi-level data set, obviously, of Sanger sequencing from different regions of the genome. We have looked at several other loci in the mitochondria that consistently show that result, although not in conjunction with one another.
Okay,
So, but I don't know if I answered all your questions. The last one, what the genetic material that these look like, these are all isofemale lines.
okay,
But these species are not friendly like melanogaster. You can't really inbreed them
Okay.
directly. For
okay,
very long.
okay, okay.
So, yeah, isofemale lines is about as genetically homogeneous as you can get.
And then like a kind of a technical anal question, when you extracted DNA from ovary for the Illumina paired-end sequencing, did you take the entire ovary or the same section of the ovary? I don't know if there are one or two.
Yeah.
It makes a difference in animals,
Yep.
is why I'm
So,
asking.
I did both ovaries. I'd say
Okay.
shortly briefly, I mean, for the RNA. check, right?
Yeah,
Yeah,
sorry, not,
yeah.
yeah, okay,
Yeah, and that, and I also included, it basically was just the reproductive tract, moving up to including.
okay, all right, cool, thanks.
Yep. Yep.
Great talk. Is co-infection with multiple strains of Wolbachia common? Because when you showed that agarose gel, it looked like
Yeah.
you saw both the deleted one,
Right,
the male killing one, and also the CI
so.
larger one.
There are instances of it, it's relatively very rare compared to single infections. So, yeah, I would have to get into the details of what those primers look like. And I tried to simplify the story a little bit. So that deleted region in my male killing Phylogeny infection, if you look, so like I said, it's flanked by transposases. And so it looks like it cuts 2 transposases in half, and then those are fused together. So inside of that deletion are the other half of that transposase. And so there is some still, I guess, homology, like I said, parallel, not homologs, but designing primers that can target one specifically, not the other, is kind of difficult. And so I basically had to look for the very few nucleotide variants in that, put those right at the 3' end of my primers. But what I think I'm getting there is amplification, both in the leading region and outside of it. So I think I'm getting that.
How does horizontal transmission occur between things that are far enough apart that they're not actually hybridized with one another? Because the phylogeny you showed in the beginning were some very divergent crosses of transfer. What's happening there?
Yeah, so the honest answer is people don't really know, but they have pretty promising hypotheses that one mode of transmission could be through mites. The mites also cover a lot of Wolbachia infections and parasitize a lot of these Drosophila species. And another leading hypothesis is parasitoids with wasps, which people have shown can carry Wolbachia on their ovipositors for limited periods of time. The problem is when you look at like that phylogeny, I mean, you could have a transfection rate that's inconceivably low to observe in a lifetime that can produce those patterns. But yeah, that's kind of the best. Yeah.
I'll ask it later.
Right,
It's gonna be a fun one.
All right, thank you. Have any questions? End the show here and say bye. We're getting some claps online there.
Very good.